Hi, everyone. Welcome to Foresight's Molecular Machines Group. I'm really excited to have so many of you here. I have an exciting announcement, which is that we have Eric Drexler, one of our co-founders, come back to the Bay Area in October, and we will be doing a launch of his new molecular machine systems engineering platform, and we're also going to have a nanotechnology workshop. I will share more info about that in the chat. But for now, I'm really excited today to welcome Dr. Lurong Pan. And thank you so much for joining. I think we're not only going to be in the delight of a presentation about what you are working on, but also a demo, which is always, I think, the most exciting part of these meetings, because it's a bit more interactive and you actually get your feedback, possibly. But yeah, you'll be today presenting on protein LLM and generative AI in protein engineering. I'm going to share a lot more about you in the chat. I'm going to share your bio and so forth. And yeah, without further ado, please take the stage, and I will be in the chat monitoring for questions, and we'll be ready in case any bits come up. So thanks a lot for joining. It's wonderful to have you here.
Thank you, Alison. Hello, everybody. My name is Lurong Pan. I'm the founder of Ainnocence, a AI platform company for drug discovery. I was a trained computational chemist and also AI scientist with about 15 years of experience using computational tool to design medicine. And Ainnocence is the company that I recently founded that leveraged the most advanced AI platform, which is underlined. It is we call the protein language modeling. And this is also being featured in Wall Street Journal, which is the same algorithm power the chatbot, which is the ChatGPT, is also be able to be utilized in new medicine discovery. And let me explain a little bit the theoretic background of how those two seemingly unrelated information can be working similarly in a way that to help in our medicine field. So a little bit introduction about our, probably most of you already used ChatGPT for quite some time and understand that it is based on we call the large language model, which is a computer algorithm that's been learning the human language and the format, and then be able to generate like sentences based on its context. This is not very hard to understand the concept. But what is amazing is after you learning so much like the large enough of database of a human language, the AI is able to not just generate correct sentence with correct logic, but be able to even inference more complicated logic. So this is basically demonstrate the potential of this algorithm. But let's take another look at our actually the composition of our human body is started from DNA and proteins, which is also sequence. And we call that protein language because it's just like a language that is encoded in letters. And it's a 1D vector with context. Even it does not have any somatic meanings, but it actually has genetic meanings and also functional meanings in chemical physical space. In that case, it's also similar that if we could learn a universal protein sequence database, no matter it's human or non-human, anything in the nature, if the database is large enough, it would be able to train the AI, be able to generate sequences that's actually similar to nature. In this case, this is basically lays the theoretic background of we can also perform the similar training and learning and inference process on protein sequence like human language and be able to predict what we wanted to predict, either generate a functional protein, such as some modified enzymes or antibodies or other type of protein species. And at the same time, we'll also be able to investigate the interactions between them by training against binding information or other type of observation that we can observe from them. Basically, that's just a little bit background. And based on that, we actually created a platform that be able to perform a generative AI task for a few different things. Specifically speaking, for those of you in biologic discovery should be very familiar with those terms. For example, affinity maturation, which means we can modify a sequence of antibody and increase its affinity towards the target. And then we can do de novo design, which means we can from scratch to design any therapeutic proteins. And we can do off-target binding predictions to predict any protein interactions, such as antibody membrane protein interactions to avoid potential safety issues and all the other protein inherent features that can be linked to the sequence. And then what we were trying to do is actually really replace the traditional way of how antibody, for example, was being discovered. And the traditional way of probably a lot of you familiar with phage display, which is a wet lab process with so many steps to generate like a real libraries of antibody sequences and then be able to screen them against certain target and with a series of steps of equipments. And it normally takes 3 to 6 months with very high cost. And actually half of the time you can't even screen some of the antibody you really wanted to against your desirable target. And another way is animal immunization. You have to inject a purified antigen to rabbits or mouse or llama to generate your antibody therapeutics. And then after that, you have to do humanization. All of that are really a lengthy wet lab process and very expensive. Using this AI platform, what we can do is we'll be able to minimize everything from time points within the months and with very low cost. So far, we have done over 50 projects, about at least 80% success rate, and we don't involve animal. What we do is we take an antigen sequence, which is the only thing that's required as an input. And then we started to either generate antibody or we generate from a template that you think is not good enough towards a KD or IC50 that you desired. So then we just started to do the virtual library building. And we can build a library up to 10 over 10 or even 10 over 25th possibility by mutation on the CDR regions of the antibody. And then we will be able to do more. I'm sorry, there's some chat. Somebody have questions?
It's all right. It's just someone joining. I got them.
Okay. All right. So let me just minimize that. And then after that, we only need to take the top ranked sequence, normally 20 to 50 sequences for expression and do a binding assay. And then we'll be able to find the antibody we desired. And what's good about this part is, for example, for binding assays, a lot of the antigens in the traditional part, if you don't purify it or express it as a whole, you can't even generate any antibodies against it. But within this method, you can directly test your antibody in a cell-based assay or even like an animal assay, so that it can. You don't, for example, we know that antibodies target mostly are membrane proteins, very hard to deal with. So you don't even need to purify that or isolate it. You just leave it on the cell and do your binding assay. So this is like a very straightforward process. So we have done a lot of those tests already. And let's introduce a little bit like that, how this software works. I will later demonstrate this software, but basically, we have done a protein large language model, which trained over 3.5 billion of sequences of like human antibody database. That's we already know, which means we equip this AI be able to generate any sequences that's first already humanized. Second is an antibody sequence that can be expressed successfully. And then we do a fine-tune model based on our domain application. For example, in this case, we were trying to optimize affinity, which is antibody-antigen interaction data. A lot of data are like SPR binding data or some IC50 data from various type of binding assays. So we have hundreds, thousands of those data points to be able to fine-tune it. And the entire process, basically, we take the sequences of antibody antigen and we converted them into some kind of matrices as like a transformer model and directly approximated to the binding score. So we have, and then what happens is the computer will basically ranking the top affinity sequences among millions of outputs. And then you only need to take the first 50 antibody sequences with those different mutations on the CDR regions to the test, and you would be able to get your desired antibody. So it's a very simple process from target sequence to de novo design. And normally we do the ultra virtual screening on the Amazon cloud. And also it's distributed computing. No matter how complicated this case is, it can be done within 1 week. So the computational time is very fast. And then currently for the protein expression and testing, if you have your own lab, it's very straightforward. And a lot of lab that does protein expression can be done within a week. Then 1 week expression, 1 week test, you'll be able to generate the data. Sometimes we need to do a second or third rounds of computation to optimize the affinity more and more. Each time you get a feedback from the wet lab, you actually fine-tune the AI model a little more and then make it fit the target profile better. We normally take 2 or 3 rounds. So that's how it works. We benchmark the software with other software that has been in the place for many years. Because there's a very drastic shift in this field, is people start to use sequence based type of deep learning approach replace the conventional 3D reconstruction of antibody antigen interaction. In the past 30 years, there's the bottleneck of protein interaction in terms of 3D structure model, because it's just impossible to sample all the configurations of 2 proteins in the water. It's just computationally impossible. Even we have a super quantum computer, is still not possible. So that's make it really hard to do a biologic design using a 3D model. It's probably doable for small molecule, but not doable for a large molecule. But we think this sequence-based model actually opens a new door and possibility. So we benchmark for example, this is a particular benchmark set for a single mutation generated delta G change for a antibody antigen testing sets. The accuracy is from 0 to 1. So basically 0.5 is random guess. Anything better than that is a better prediction. Our system actually outperform all the other structure-based software and for known target, which means it's in the training set, or for the new target, which is not in the training set. There's some differences between the target has been seen or not been seen, but still the score is much better. And what's more important is the computational cost. The reason we can't really virtually screen a large molecule because, for example, our B cell generates 10 over 10 to 12 of possible antibody when we get infected. If we wanted to mimic that process, we have to generate as much antibody as possible virtually. And in the past, all those software like Schrödinger, they're stuck on this 3D generation. So even right now, there's AlphaFold 3. They can still generate 3D complex, but still the time cost to do that is just really big. So that, for example, if you want to generate like 10,000 of them on a single GPU, it take 100 days. It's just impossible to really sample all the sequence space. In that case, we use sequence is very lean model, which dramatically reduce the computational cost by about 10,000 times. So we'll be able to really finish all the tasks within 1 week. So then I just briefly introduced some case studies, just shrink a little bit to different size. Okay. For example, first we do affinity maturation, which we basically just mutate the CDR regions for heavy and light chain of a template antibody you think is not good enough a binder. And then we can do this for one target, or we can also do it for multiple target. We have done for example, infectious disease. We normally do COVID, flu. We do over 50 antigen at a time to make sure the antibody is broadly binder. And then for other human target, we do selective binder as well. But for example, this is EC50 result for the binding. The lighter color is the stronger binder. For the first round of computation, we recommended about 20 antibody based on modification of the templates. And then we get about 40% of them are better than that. And then we do another round after retraining the model. We get a better profile. about 10 to 100-fold increase on the binding. So this is like a very typical case of affinity maturation. Instead of using wet lab method in the past, this is just very easy and lean methods. We can also do de novo design. So we normally start with a diversified generated antibody, and then against a certain protein, and then we start from finding a weak hits, and then we expand the weak hits to more similar sequences and be able to identify the desirable one. And you can engineer any form of antibody you want. The AI will generate it very easily. And we can also do epitope mapping. So this is almost like a back scoring. Instead of scoring different antibody against one target, we're scoring one antibody against different epitope. And then we give the scores and then be able to rank them. It's also very straightforward. And then we also developed a comprehensive development score system, which considered some of the unwanted motif, easily digest unstable motifs, and isoelectric scores and thermal scores, and be able to design the antibody at the beginning or even before the development stage quite easily. And then humanization we also developed the similar approach. We trained the sequence of all the human protein sequences and antibody sequences against all the non-human species, be able to find any given sequences of peptides or antibodies that has how much humanness it has. And then we'll be able to modify, for example, if your starting point is an animal antibody, and we'll be able to humanize it for you as well. And the last one is also very useful. We find out is like the off-target, because a lot of clients of us are concerned about either the weakened efficacy of the antibody or some unwanted side effect is from the off-target effect. And off-target is really hard to screening by wet lab because you really have to really I think collectively our membrane proteins and our secreted proteins has about 6,000 species. We really have to, if we have to screen them, even in a library or panel setting, is still very expensive. But here it's just one click away. Yeah, so far we have served the clients globally with universities and different pharma companies, trying to target the AI service market right now. But we try to collaborate with different companies that related to protein generation, no matter it's just therapeutic protein or industrial enzymes in synthetic biology field, or some of the biomass from renewable energy space we can also do. And this is our team. This is me that have 2 degrees and with 14 years experience, and we have 2 very experienced biomedical scientists and AI scientists, and we have Dr. Ding and Dr. Tanik as very experienced professor in the biotech and AI space. So that's the presentation part of my talk, and I will stop sharing this and jump to the demo. So this is completely native on the cloud, but if the client need, we can do a local deployment. So this is the different functional module I mentioned earlier, such as affinity and some protein designs and humanization and some off-target. So I can just show quite quickly how a task is being set up. Sorry, let me drink some water. Basically, you can just simply input the sequence of your, for example, if you have a template, you want to perform affinity maturation, and then the antigen sequence, which is could be for membrane protein, could be an entire extracellular domain, or if you know the epitope, it could just be just a small peptide. And then you can choose you wanted to, of course, increase affinity, or if it's unwanted off target, you wanted to decrease affinity, and you can do different search algorithm. It's just a different speed of how it will be finished the task. And then after you submit, it's automatically sent it to the cloud, and we have everything set automatically. And then you can download an Excel file, or you can just visualize the results like I showed earlier. Just earlier, those are the mutations the AI recommend that has increased affinity dramatically. And then you can just take the sequence for your experiment. Yeah, and for the fusion protein engineering engine is the same. For example, if you are not a standard antibody, it's just some peptide or some nanobody, a single chain, you can just put a protein in it, and then you bracket the area you're trying to do mutation on, and then also the target sequence, and then you choose the algorithm, and then it send it to the domain. And then also it has a downloadable sequence. You can do the next step. And for the humanization, it's, for example, if you have some kind of mouse antibody sequence, you can put it on it. And then after you perform the humanization task, it basically will mutate the sequence right here and also show you the score before and after it's being mutated and how much the humanist score has been changed for that particular antibody. Yeah, and off-target is also very straightforward. You put the antibody sequence and then target sequence, and you can also upload a bunch of them. And then in the end, then it will show you some of the alert score for potential off-target risk. Yeah, so basically it's very simple to use. Hopefully it's much better than the computation kind of software we used to use in the past. And it's just designed for biologists that even without any IT training, be able to use it. Yeah, so that's all my talk today, and welcome for any question.
Fantastic. Thank you so much. I know that maybe I'm gonna ask my questions in the end because we have so many other questions. Are you okay with taking a few questions from the chat? Then I'm going to ask people to unmute. And the first one would be Samar Sandeep. If you want to unmute to ask a question, go for it. Otherwise, I can read it out from the chat. But you're first.
Awesome. Yeah, thanks for the presentation. Great to learn about AI Innocence. I guess my interest was in the biomass focus. I definitely have seen DOE focus in this area. And I myself am also run a company in a similar field as yours, and I've tried to look at that. But I didn't quite understand where protein interactions or protein ligand interactions would really work there in biomass, because you're dealing with proteins on proteins. So I'm just curious where you were seeing your technology potentially being helpful in biomass and renewable energy.
Yeah, thank you very much. I think there's probably two part of it that can help. First is I think there's a lot of bioreactions related to generates biomass that's involving enzymatic catalytic process that we can actually engineering novel enzymes that generates some of the downstream material more efficiently. And then another is, for example, if it's just to generate certain material itself, if you have a certain energy profile of your material that has historical data we can train on, we can definitely also let the AI design accordingly or modify the current materials, if that makes sense. But I think it's probably, I don't have a case study right away in my mind. But if you have any like a particular case, we can probably discuss more in detail.
Okay, thank you.
Wonderful. Cool. Next one up, we have Paritosh, and then we have Micah.
Hey, thank you so much for the presentation. It was really exciting. My question is mainly around your enterprise clients and what value they find most in the solution, like where they saw like the most difference it made.
I'm sorry, I didn't quite understand your question. You mean what part of the function that has the most commercial value? Is that what your question is, or?
I think so, something like that. He also asked in the chat, who are the clients in terms, who are the clients and where did they find the most value? This is trying to scout out a few user profiles.
Yeah, sure. Definitely. Biotech is our biggest client. And the biggest value is that save their R&D costs and time. Yeah, because they're trying to race against their peers for certain targets, or they have very limited budget to set up a traditional high throughput screening platform. So this is like a very economic jumpstart for their projects. And another one is our CRO company. Because CRO company, they're serving a lot of different pharma and biotech. They need to like scale in a scalable way to do it. And we can deliver what they want very quickly and scalably. In the past, computational work normally takes 2 to 5 months for a project for a drug design work, but now we just take a week, so it can be easily scaled up.
I think he was clapping, which means to me that you said it thoroughly as it is right. Next one up, we have Micah.
I have a few questions, so if anyone will raise their hand or feel free to interrupt me. First one off, in your demos, you were basically modifying existing antibodies and whatnot. Do you also have the capability right now to do novel antibodies? So where all you have is a protein and you want to find an antibody for it, you know nothing else? Can you start with that, or do you need to start with a template?
Yeah, so basically for the front end, we have a template to start with, but basically when we don't have a template, we will use the AI to generate it, and the back end with the reference of a human antibody database. So that's how it works. Yeah.
And can you generate catalytic antibodies, or do you not have enough catalytic antibodies in the training data set to actually narrow down to just that?
You mean catalytic antibody? I don't quite get.
Catalytic antibody is just basically an enzymatic antibody. It's an antibody that, when it binds, instead of just binding and sticking on the target protein, it actually breaks it in half. Useful in biology when you have a protein that's in circulation, you want to clear it out, and so you always want to get rid of it all. The big advantage is if you have just regular antibodies, you need like 1-to-1. So for every protein you want to remove, you need one antibody to flag it for the immune system. Whereas with the catalytic antibody, one catalytic antibody can clear out thousands of the target protein. The problem historically has been your body sometimes makes catalytic antibodies, but like very rarely. It's like dumb luck. Sometimes you get lucky and the antibody happens to be catalytic. And when that happens, it's great. And we find a human that happens in, and we harvest them, and we then go mass produce those. But it's very rare. And so I'm on the lookout for any mechanism we can use to generate these more intentionally, because they're really powerful tools for biology and vaccination and stuff like that.
Yeah, I think it's doable as long as you're sure about the action of the mechanism of action. For example, you have the target of 2,000 proteins to be bind to in a very specific binding affinity range. I assume it should not be too strong or too weak. And then we can design antibody just fit that profile and be able to serve the needs. Yeah.
Okay, I'll let Ted go.
Thank you.
Yeah, Ted, feel free to take over.
Okay, so I don't quite understand how your LLM is trained. Are you training it on pairs of antibody and antigen that are known, or is it just the sequences of the antibodies?
Yeah, there are 2 steps like I showed in one slide. So the first one is pre-train, which is just on the antibodies. So make sure the AI extract all the antibody features correctly. And then we train the pairs, antigen antibody pairs, to make it more directional. Yeah.
Okay, good. Yes. And then when you're asking it, you effectively can just ask, here's the antigen, generate an antibody from a known one that will attach to it. That's right.
Yeah. Mm-hmm.
Neat.
Thank you.
Cool. I don't know, Micah, if you have any follow-up questions, or anyone else, if you want to raise your hand, feel free to go for it.
One kind of big question. If you had to guess, the database of human antibodies that you're using, do you think that you could have achieved similar results with a significantly smaller database, or do you think that you could receive significantly better results if you had a bigger database? Or do you feel like kind of the database size is just right? Like where do you think we are in terms of volume of data versus how effective the training is? Do you think you could do better with significantly more, or is it kind of marginal gains at this point and you're already long past?
Yeah, that's a very good question. We actually tested that by increasing the numbers, and it's reached a plateau when it's at the 100,000. So
Interesting.
it's very interesting. We wanted to know if we increase, but anyway, but it doesn't really change the performance after that for some reason. Yeah.
All right, cool. If anyone else has any other questions, please let us know. I have a few questions that people always are interested in, usually when it comes to new breakthroughs and especially projects that are out and about and to be used here already. So one question, for example, let's start with here. I wasn't quite able to find the page. I was only able to find this one here. If people want to sign up, would they contact you?
Yeah, our
or would
website
they-
is in maintenance today.
Got
Yeah,
it.
maybe if you can just email me, that would be nice. Yeah, I can send you all the material of all everything and the presentations and our contacts and everything. Yeah.
Okay, good. I just want to make sure that I'm on the right page and so forth. But yeah, this will also air on YouTube a bit after the actual recording. So at that point, it's hopefully all good again. Okay, Micah, you had another question. Go for it.
Yeah. How far off do you think we are from being able to generate enzymes that produce a desired reaction without any existing examples to build off of? So let's say you just want you have two molecules and you want an enzyme that makes them bind together reliably. How far away are we from being able to describe that interaction only and then say, make me an enzyme? Do you think we're close, or are we still a long way off?
Actually, I think there's already some publication from David Baker's lab that some completely artificial proteins have some kind of enzymatic function. Yeah, he's in the field for much longer than me, so probably have more to see. But he's focused more on the 3D generation. But definitely, I would see the potential of AIs in the protein spaces because the interesting is because human language has so much diversity of words and things, but proteins only have 20 letters. Basically, make it very easy to generate functional but novelties in a very limited sequence space. For example, 500 amino acid. There's just a lot of rooms for innovation. And I think all the data we have currently available in the field are probably already enough to achieve that. It's just the tweaks on the algorithms and design workflows. Yeah.
Very exciting. Thank you.
Mm-hmm. Yeah.
All right, cool. I'm going to come in with my standard questions until anyone interrupts me by asking another question. Feel free to do that for sure. I guess maybe building up a little bit on my guest question just now, are there other things that you could see being possible in the next 5 years? Like if you just continue to develop the system as is right now, like where do you think on a speculative hunch we could be in a positive world? If you continue to move forward, make progress, what's possible in terms of like applications that might arise from this or in terms of entirely new discoveries that could come out of it?
Yeah, that's a good question. Actually, we're currently testing on the CAR-T technology using this platform, which means we can basically engineer any type of CARs on the T cell. And it's similar. Yeah, it's just we just basically design the protein, but we just, back translate it to DNA sequence and put it back in the cell. So in that case, I think, I feel like this is a great potential in the cell biology field that we can almost program the cells the way we want and make them go anywhere we want them to go to be very highly specific. And then we can modify anything on the surface of the protein that they used to have or they don't or whatever. So I feel like it's just really opens a new door for cell engineering as well. But I understand there's more complexity involved, but that's the potential, I would say. Yeah. We're still testing the T cell platform. We haven't have the successful case yet, but we hope we can have something soon released.
That'd be fantastic. We have Ted with another question.
Yes, thanks. A really interesting application here. So one of the things that is common with large language models is what is called hallucinations, or that they can be very confident about an answer, but it's completely bogus. And I'm wondering, in your context, do you have any examples of that, or where the system will say, I just don't know, as opposed to make up a bad answer? Or is that not so much an issue in your application?
Yeah, I think for this, because like I've said, the letter space is so limited. It's just 20 letters. And then the only output is just it's better or worse towards a particular function we try to predict. So when it's just, but of course, there's a lot of false positive. that's been generated. That's why we only have about 20% hit rate. 15 antibody, we get 5 or 10. That's correct, but the other incorrect, which means all the incorrect ones are false positive. But those are things you can't really avoid in the sense of data-driven methodology, because still in the training set, there's very huge amount of errors and incomplete information, and that could potentially cause this issue in the downstream prediction.
Yeah, I'm wondering of whether this might be then a more advanced technique eventually. If some question is so far away from the training data, it somehow would know that it's basically extrapolating instead of interpolating in sort of the old statistical lingo. If it could make that this distinction, but I guess that's not yet part of what these models know how to do, I guess.
Yeah, they could have some ability to do transfer learning. When, for example, a target that has never seen and also it's very different from the training set, but it will reduce its accuracy. But because it has some kind of this ability of transfer learning, it will be able to work on the new targets. Yeah.
Good.
Oh, Micah.
So one of the problems in biology, as most people know these days, is the immunogenicity, where you have some protein that does amazing things, but you can't put it in a human because it'll kill the human because their immune system responds to it. And a part of this is, as I understand it, is a humanization process, right? Is that process alone, like the humanization stuff you have, is that alone enough to take some protein from like GFP, humanize GFP, so you can start making humans glow? Or do we need to do something more to make it so arbitrary proteins can be put into a human? And if so, do you have any thoughts on what the path looks like to getting there?
Yeah, that's a good question. So we have in the past we have tried 2 versions of humanization model. One is we based on the HMC 1 and 2 families that make sure, and we cut all the proteins in like sequence of 8 to 12 amino acid and be able to predict the binding of that. So that's basically where the immunogenicity comes from, right? And then we have done one of that model, and then the second model is actually has nothing to do with biology process. It's just to simply distinguish between human and non-human species by simply training on their sequence. And we find out the performance of the latter one is actually much better. So in that case, I can't really see in different type of scenario which model would perform better. But I think it's, for example, if you generate a random protein and then we're using that humanized model to score it, and if it's past the score, if it's past that threshold, we would probably think it has no immunogenicity when injecting it. So that's, but we still need a lot more testing done on that side. But both ways we can use computational model to address the immunogenicity.
Cool. That was clearly quite exciting. If you think a little bit ahead in terms of if we don't get there in time, what's the current bottleneck that's blocking progress? And that could be, or I guess it's partly data, probably. Are there any specific things that like you're really like bottlenecked by right now? And then I guess relatedly, if someone new is entering the field right now and really wants to make progress, are there specific challenges that you'd point people to where they could actually be helpful and can like unblocking progress a little bit? Like some bottlenecks are just like cost of compute or something that's like harder to solve for some people in the field. But
yeah, interestingly, the biggest bottleneck we got is not data or computational power like most AI company. Our biggest bottleneck is the validation time to do the wet lab is too long, which means we are not fully occupied. We have to do a lot of business development to fill the sales pipeline. Since we do one computation, probably finish in a couple of days, and then we have to wait 2 months to get an experiment result. And then until I finish 1,000 project, people probably won't be convinced to adapt as an industry standard. So we will have to, and I don't know, I can't see how I can expedite this process. We wanted to serve different needs and diversified profile of biotech that work on different target profiles so that we will know how universally applicable this technology is. But we still have to wait the wet lab result. And we have to wait their get funded sometimes. So We have to evolve with the industry together. I would just say that. But I think, yeah, if we can get more capital, we probably can do more of an internal test and to just do the wet lab ourselves more efficiently. Yeah, that's kind of
So with the follow-up,
Relating to that, so you mentioned that the run times for your stuff is on the order of hours to like maybe a week, depending on what you're doing. Is that like hours to a week on a single GPU, which is relatively inexpensive, or are we talking a cluster of 100 H100s and it's costing you $1,000 an hour?
Mostly single GPU. Yeah, we only
Okay.
do parallel when we do training test. That's why how lean this model is. Because inference doesn't really cost that much computational power. And when we even do training, we normally train only when the second and third round is just only a few days on maximum 8 GPUs. So just one.
Okay, so the operational cost for actually generating data is relatively inexpensive.
Yeah.
Operationally.
Yeah, the cost is still on the wet lab side. Yeah.
and if you think about just what's ahead in the next few months for you guys, what are you hyper focused on right now? What's going to come out soon? What can people get excited about in the near future from you guys' team?
Yeah, we're trying to actually, that's why our website is down. We try to create a user portal so that everybody can sign up and try it on it. So we're doing the engineering right now. Yeah, hopefully get released in a couple of months. And then, of course, we're trying to serve more project, actively develop partnership and expanding in different area. Like I said, the CAR-T space and other. Yeah.
That's exciting. Mike, I had a follow-up. Go for it.
Yeah. On that, is your target to make I don't want to say direct to consumer, but direct to user? Most LLMs, you just go onto a website, you put in your credit card, and you can start using it, right? Is that kind of your target, or is it more building like a B2B sort of thing where you've got like a sales call and go through a bunch of rigmarole, and then eventually you can get access to something for a while? What's your who are you targeting demographic-wise for your application?
Yeah, good question. So we're currently doing the latter one, like a B2B, but I found it's not very efficient. That's why I wanted to try the first model. Even I know in the scientific field, people tend to have a lot of more conversation before making any decision. But I think as long as our price is reasonable, it should be okay. That's just having a human AI interface to finish the process. Yeah.
Awesome. And then the final question from me would be that if people here on this call or afterwards that perhaps see this video on YouTube, get excited about your work and would love to support you, are there specific bottlenecks like for you specifically right now? Are you trying to hire? Are you fundraising? Are there any specific collaborations you're seeking out right now? Anything that people can help you with at all would be useful to know to close it out.
Yeah, we're always trying to raise money. Yeah. So yeah, I'm currently work with a startup fundraising agency trying to open up almost like a crowd raising mechanism so that more people can participate. This will be released pretty soon. Yeah.
Okay, cool. How can people learn more about you? I know that you have a Twitter handle, right? That was posted, but if people just want to follow you, stay up to date, and maybe join
we have
fundraise-
linking. Yeah, we have the LinkedIn profile for the company. Yeah, and also myself, and then I will start to release the links. Yeah.
All right, great. So then I guess I would direct folks to LinkedIn, and if someone is really excited about speaking with you, please just contact me, and I'll try to get a hold of you.
You can all find me on LinkedIn. Yeah, if you want. Just invest.
Okay.
Yeah.
Cool. Thank you so much. I really appreciate you coming on and not just like presenting the work in theory, but actually giving us a demo. And yeah, we're really excited to have more folks in our community try this out. And thanks a lot for your work. It's very exciting.